Welcome to Computational Biology. Today, we are diving into a crucial statistical tool called the coefficient of variance. As biologists, we constantly measure things, from the size of microscopic cells to the weight of massive animals. But how do we compare the variability of completely different biological traits? By the end of this video, you will understand exactly how to measure and compare relative variation, ensuring you are never misled by raw numbers again. Let us start with a thought experiment. Imagine you are studying a massive African elephant and a tiny field mouse. Suppose you observe a weight variation of exactly 2 kilograms in both populations. For an elephant weighing thousands of kilograms, a 2-kilogram difference is absolutely nothing, barely a rounding error in its daily food intake. But for a tiny mouse, a 2-kilogram difference is physically impossible and would represent a monstrous change in body size. This highlights a major problem in statistics. This scenario perfectly illustrates why simply looking at the raw spread of data, like the standard deviation, does not tell the whole story. When the baseline sizes of what you are measuring are completely different, absolute variation becomes misleading. We need a way to measure relative variation, a tool that tells us how big the spread is compared to the average size of the object we are measuring. This exact need is why mathematicians and biologists rely on the coefficient of variance. Now that we understand the problem, let us look at the solution. The coefficient of variance, often abbreviated as CV, is a statistical measure that represents the ratio of the standard deviation to the mean. Unlike standard deviation, which keeps the original units, the coefficient of variance is a relative measure. Because it divides the spread by the average, the units completely cancel out. This leaves us with a pure, dimensionless number, which is incredibly powerful for comparing completely different data sets on a standardized scale. This dimensionless nature brings us to its real-world importance in computational biology. Imagine a geneticist comparing 2 genes. One produces thousands of transcripts, while another produces only a handful. If we only looked at standard deviation, the highly expressed gene would almost always look more variable, simply because its raw numbers are larger. By using the coefficient of variance, researchers normalize the variation against the average expression level, revealing which gene is truly fluctuating more relative to its normal state. Without this, biologists would constantly misinterpret data. This brings us to the mathematical representation. The formula for the coefficient of variance is beautifully simple. We take the standard deviation, which represents the absolute spread, and divide it by the mean, which represents the central anchor point. In this formula, sigma represents the standard deviation, telling us how much data points deviate. Mu represents the mean. By dividing sigma by mu, we find the fraction of the spread. Multiplying by 100 converts this into a readable percentage. If standard deviation increases while the mean stays the same, the CV increases, showing wider relative spread. Before we move on, let us see this with a concrete example. A computational biologist is analyzing 2 genes. Gene A has a mean expression level of 5,000 transcripts with a standard deviation of 500. We want to find its relative variability. We can't just look at the raw standard deviation. Instead, we divide the standard deviation of 500 by the mean of 5,000, giving us 0.10. Multiplying by 100 gives us a final coefficient of variance of 10%. This means the typical variation is about 10% of its average level. Now, let us look at the second gene in our example. Gene B has a much lower mean expression level of 50 transcripts with a standard deviation of 20. Again, we apply our formula. We divide the standard deviation of 20 by the mean of 50, which gives us 0.40. Multiplying by 100 yields a coefficient of variance of 40%. Comparing the two, even though gene A had a much larger absolute standard deviation, gene B is actually far more variable relative to its baseline. Its activity is much more erratic and fluctuating. This ability to compare relative spreads leads us to another incredible use case, comparing entirely different biological units. Suppose a zoologist is studying adult male lions and wants to compare body weight in kilograms to tail length in centimeters. This is the classic apples and oranges problem. You cannot compare kilograms to centimeters using standard deviation alone, because standard deviation keeps the original units. To make a valid comparison, we must strip away the units entirely by calculating the dimensionless coefficient of variance for both physical traits. Let us calculate this for the lions. The zoologist finds that the average body weight is 190 kilograms, with a standard deviation of 15 kilograms. We take the standard deviation of 15 and divide it by the mean of 190. Notice that the kilogram units on the top and bottom cancel out perfectly, leaving a pure number. Multiplying the decimal result by 100 gives us a coefficient of variance of approximately 7.89% for the body weight. Next, we do the exact same process for the tail length. The average tail length is 90 centimeters, with a standard deviation of 12 centimeters. We divide 12 by 90. Just like before, the centimeter units cancel out completely. Multiplying by 100 yields a coefficient of variance of approximately 13.33%. Because 13.33 is significantly higher than 7.89, the zoologist can confidently state that tail length varies much more from lion to lion than overall body weight does, despite the different units. Now that we have calculated these values, let us visualize what relative variation actually looks like. On screen, you are seeing a graph with two very different bell curves plotted on a measured value axis. The tall, narrow curve on the left has a small mean of 10 and a tight standard deviation of 2. The short, wide curve on the right has a much larger mean of 100 and a wide standard deviation of 20. Despite looking completely different in absolute spread, both curves have the exact same coefficient of variance of 20%. The diagram perfectly illustrates how variation scales with the mean. To wrap up our lecture, let us review the most important ideas. The coefficient of variance is your ultimate tool for comparing the uncomparable. We learned that whenever you evaluate the spread of data across vastly different scales or units, standard deviation alone will mislead you. By dividing the standard deviation by the mean, we create a standardized percentage-based measure of relative variability. Mastering this concept ensures that you will never be tricked by large numbers when analyzing complex biological datasets. You now have a solid understanding of this essential statistical tool.